Cool.
Mutations refer to a process of making a mistake in the coding region of a strand of DNA. That is a gene. The sequence that would normally code for a protein is flawed somehow. Mutations can occur spontaneously, or they can be induced by chemical or physical agents known as mutagens, like X-rays or cigarette smoke or industrial cleaners. There are two basic types of mutations. There are gene mutations and then chromosome mutations. Gene mutations are the most forgiving of the two. They usually involve only a single gene. Typically they occur during replication, when the DNA is separated during the S phase, and each half is rebuilt using DNA polymerase. There's a mistake, and the mistake can take two different forms. They can either result in a point mutation or a substitution, when rather than placing a T, an A is placed instead. Simply a letter is misplaced in the original code. So that means you have one base pair which is off. That is a substitution. That may not make any difference because there are several different ways to achieve the code for a particular amino acid. So it could be that it's innocuous, or it could be that it creates a minor defect in the protein, or it could be terminal or fatal. The other type of mutation is called a gene mutation is called a frame shift mutation, and it's caused by either a deletion or an insertion of one of the base pairs, again, typically during replication. For instance, the substitution may look like a strand going from TATCG to one going from AATCG. Only a single base pair has been changed here. A frame shift mutation involves inserting a letter that doesn't belong there at all into the middle of a code. And what that does is it throws the codon frame off. So every downstream codon is now different than it originally was. For instance, here is a strand of DNA, ATACCGTAG. If I inserted an A in between the T and the A located here, my new reading frame would be ATA, ACC, GTA, and then G. So every frame or every codon downstream of the insertion is incorrect. This is a very serious type of mutation. You can also achieve the same by removing a letter. The frame shift again occurs, and your reading frame of every downstream codon is completely different. A chromosome mutation, there are two different types. One of them occurs during crossing over, usually in prophase 1, and they can be either duplications, deletions, inversions, or translocations, and I'll go over those specifically in a second. Or you can have non-disjunction, which we covered in a previous chapter. That usually happens because of spindle fiber errors during separation of chromosomes during anaphase 1 or anaphase 2, so that rather than the homologous pairs or the chromatids going in opposite directions, they both go in the same direction. So one gamete ends up short and the other gamete ends up with too many chromosomes. That's non-disjunction. We'll discuss now crossing over errors that occur during prophase 1, namely duplication, deletion, inversion, and translocation.
Action.
Deletion is a chromosome mutation caused during crossing over prophase 1. When you have two homologous chromosomes line up and exchange segments with each other. In this particular example, we have one homolog, which is capital A, B, and C, and another one which is homozygous for the recessive, little a, little b, and little c. They line up and they break off at the C and they switch. The little c is transported over to the homolog that was capital all the way through, and the large C should be transferring over to the homozygous recessive chromosome, but it gets lost. And so this chromosome now only has an A and a B, and it's missing the end caused by an error during this crossing over process, and this is known as deletion.
Action.
Another type of chromosome mutation is called duplication. This is when the homologs line up next to each other again. In this instance, again, the C is going to change positions from one homolog to the other. One breaks off, and begins moving over to the other. But before the capital C breaks loose, the little c also attaches. So now we have a chromosome with both a big C and a little c, 2 C genes as a matter of fact. This also results in deletion to the other chromosome that lost one but didn't gain one back. So this is a good example of duplication. Another crossing over mistake is known as inversion. This is when chromosomes again line up next to their homolog and they separate. In this instance, C and B will separate, or B and C rather, will separate and change positions from one homolog to the next. In this instance, however, when B and C separate from the recessive chromosome, they flip upside down. And so instead of having a sequence of A, B, and C, we now have a sequence of A, little c, little b. This one evidently did correctly. You have little a, then large B, and large c. So this is an example of inversion. Finally, the last type of chromosome mutation is called translocation. This is when there is an improper crossing over that occurs between non-homologous pairs. In this instance, we have two pairs of chromosomes, A, B, and C, represented by a dominant and then a recessive homolog. And we also have another set of chromosomes, D, E, and F, getting ready to go through the prophase 1 crossing over process as well. But what happens in translocation is, as D, E, and F begin their process of switching, and A, B, and C begin their process of switching, the piece of chromosome D, E, and F that was going to switch with its homolog actually drifts into a different homologous pair and attaches to a non-homolog. So now we have a very inappropriate construction of a chromosome that contains genes from non-homologous pairs. So rather than having A, large B, and a little c, we have large A, large B, and a large F because of this breakdown here. This one, as it turns out, did not cross over, so it's normal. This one didn't cross over, so it's normal. And this D, E, and F no longer has an F, so this represents a deletion. It would be very rare for there to be a perfect crossing over between two homologous pairs, but in this instance, this sequence came up to this pair, and this pair simply, this sequence simply got lost in the mix-up someplace in the chromosome mutation called translocation. RNA processing is actually a step in protein synthesis between transcription and translation, between the making of a transcript, messenger RNA, and it being translated in the ribosome. In processing, once the DNA has been transcribed, it rewinds and the transcript remains in the nucleus, and it is attached to it are caps and tails, which enable it to both be transported to the ribosome and also control it in various ways of regulation. Something else that can happen in processing is the transcript itself, the messenger RNA, can be edited. So you actually change the code from what you transcribed into something completely different that you will translate. It's a way of getting additional information into a single gene. This gene could have been read A, B, and C and transcribed that way or translated that way. Or by removing the B, A and C can translate to a completely different protein, all using the same gene. In this instance, what happens is the transcript, the messenger RNA, is looped, and then it is cut along the base of the loop, and A and C are glued together, and then they leave and go to the ribosome where they are translated together. That is called an exon because they exit the nucleus. B, which is now waste material, stays in the nucleus and is known as the intron. It stays inside, and those materials are chopped up and recycled. This way, I can turn a gene ABC into a gene AC without having an additional gene required somewhere else on the DNA.